Let's move on to the next session. I'm Mizan, the Director of Data Science at the National Institute of Drug Abuse, or NADA. I was also overseeing the Brand Initiative Data Science program in past several years. In the following keynote presentation, Dr. Stein Aerts will talk about training sequence-to-function models on single-cell brain athletes to decipher cis-regulatory logic and design cell-type-specific enhancers. So Dr. Aerts currently heads the Laboratory of Computational Biology at KU Leuven University at Belgium. His lab focuses on deciphering the genomic regulatory code using a combination of single-cell, machine learning, and experimental approaches. His lab has developed a suite of methods for analysis of single-cell gene regulatory networks and design of synthetic enhancers for gene therapy. So with that, let's welcome Stein for your presentation. You have about 70 minutes. Then we have 2 or 3 minutes for question and answer. Stein, take away.
Thanks for the opportunity to participate here in the workshop and to present. It's a pleasure, and thank you also for all the fantastic resources and atlases many of you here are generating and building, which is really invaluable for the community, certainly for training machine learning models. So I will talk today a little bit about some new work on some new sequence-to-function models, some of it unpublished also on brain atlases, to decipher the cis-regulatory code. And this cis-regulatory code in the genome sequence underlies basically the spatial and temporal expression of genes that we can now so beautifully measure in spatially and with single-cell transcriptomics, like in the fly brain, a chicken brain, a mouse brain, a macaque brain, any species brain, as seen today many times. And so to approach this, we and many others, and also Bing Ren talked about this, we are training sequence-to-function models that are trained on single-cell atlases, single-cell expression, single-cell attack to predict gene expression from the sequence and hopefully extract from these models insight on how each gene is regulated. Basically, we would like to find where all the regulatory sequences are, enhancers, promoters, where all the binding sites are of transcription factors, who binds there, who's activating, who's repressing, how do these factors cooperate, and how do multiple enhancers cooperate. All a little bit in a dream, but things are approaching very fast, and I'll show some progress towards this, but clearly this is the future. And while we are moving towards this dream, I think together with machine learning and data atlases, it's interesting to think about metrics at different levels, like how good can these models predict enhancers and predict gene expression? How good can they assess the effect of a variance? This can be individual variation or differences between species. How good can these models design enhancers that are cell-type specific or cell-state specific? And how good can these models explain how things work? And so what we've been focusing on a lot are models that are relatively small, convolutional neural networks that are trained on single-cell attack atlases, that are local models that take the sequence of the peak, let's say, as input and are trained to predict the attack peak height. And from these, we can derive the importances, the contribution of each nucleotide, which often fall in these motifs in blocks, which we try to assign to transcription factors and thereby decode how this may work in the cell. If we also link this to gene expression and predict gene expression, then the input sequence has to be larger, and then we are in the realm of the larger models, including also transformers like AlphaGenome and Borzoi Prime, for example. One example of such a model on a brain is the first one we did on a brain was the fly brain. This is a single-cell attack atlas of the fly brain with many differentially accessible regions, so exclusive attack peaks per cell type, on which these models are trained. And from there, we can derive for many cell types combinations of transcription factor binding sites that we put in code tables. So combinatorial patterns of activators, also the absence of repressor sites for many different cell types. And in the fly brain, we can, of course, test these enhancers by enhancer reporters using transgenic flies, where this yields a more than 90% success rate. So this is really a very good approach in the fly to find cell type specific enhancers. This led then to, since these models were getting better and better, to the design of enhancers according to different strategies. For example, evolving a random sequence to a particular cell type by iterative mutations that are selected by these sequence of function models, or implanting motifs at positions that the model chooses, or generative models. And again, we can test these synthetic enhancers in the fly. Starting from the same random sequence, we could evolve it either to a neuronal type, Purkinjean cells, or a glial type. And these work also with a very high success rate. We were able to design synthetic enhancers that are minimal enhancers, so very short, 41 base pairs that work as well as 500 base pairs, and dual code enhancers that work in 2 cell types, or that the model can actually dissect out and remove or edit the sequence so that 1 cell type is removed from this enhancer and it's only active in 1 of the 2 cell types. After designing an enhancer from a random sequence here to this minimal enhancer, we can also use the model to destroy it again. In this case, we avoided all the nucleotides that are these activator sites, and this is still possible by including mutations that generate these repressor sites, and this can be dominant, so this enhancer does not work anymore, even though the activator sites are still present. This is now all come together in a package called Crested that is available. It's hopefully soon accepted. We are still waiting for the last steps. In this package, you can analyze single-cell attack atlases and do all the normalization and the pre-processing, train models in very different ways, and use downstream functions to scrutinize this logic and to design synthetic enhancers. And there's many models available, including many brain models, some of them developed together with the Allen Institute. I will refer to that later. Here's one from Bing Ren's data set, single-cell attack of the whole mouse brain. These models are scalable, so they can be trained on the whole mouse brain. And here you can see for all the different cell types at this level here included, that at the diagonal you have this high correlation between the actual peak height and the predicted peak height. And this is true for many, many, many cell types, and also cell types that are similar in attack, or also similar by sequence prediction. So this is working, and we can take these models now to validations to test to see if they can predict cell type specific enhancers, which we did in a very interesting collaboration with many of you here in the meeting, in the challenge organized by many of you with Nelson and Trygve and Niklas from my lab participated also here. And we could find that these crested models here in the blue line can actually predict on-target cell type specific enhancers with still quite some limitations. So it works better than H3K27 acetylation, almost as good as the differential attack itself. Combined with attack, it gives the best performance, but it's a good metric, and we can use these resources with all the AAV tests as a metric to further improve these models. Nevertheless, what these models provide is insight into the sequence logic, and sometimes this can be really, really robust. This is an example where a mouse model trained on the whole mouse brain is used to decode, let's say, decipher the fire enhancer, which is only accessible in microglia. And then we also give this mouse enhancer to the human model, which has not seen the mouse genome or the mouse atlas. It's trained completely independently on the human model, and you can see that the attributions found for this mouse enhancer by the human model are almost the same, almost exactly the same as what the mouse model provides, showing the robustness of this deciphering. And we exploited this further in a study where we did this correlation, let's say, between the attributions for all the enhancers of a cell type, and that for all cell types of the chicken brain versus the mouse cortex. And this finds, besides the non-neuronal and the interneuron similarities, also some of the glutamatergic neurons that share similar regulatory logic. So this is here the correlation between explainers, and we can have other metrics like the transcriptome comparison and other metrics that we derive from these deep learning models that can be used to compare cell types between species. Even excitatory neurons in the bird brain, which does not have a layered cortex, have similar logic for some cell types to the layered cell types in the mouse cortex. And this chicken enhancer that we tested here indeed is predicted and validated to work, to be interpreted correctly by the mouse cells in the brain. Crested can then, as a last step, evolve these enhancers, random sequences to targeted cell types, and you can choose any cell type that the model has learned well. And this can be done for any cell type basically in the brain and can be tested if we want to do that. That's why we are so far with sequences, the sequence of functional models, and then the attributions. And now we are working on how to scrutinize and get more mechanistic insight. And an important novelty here is a new method that we developed to decompose this motive neighborhood after we have this model trained and used it. And an important innovation here is that we represent each of these seclets, each of these potential binding sites in a high dimensional space so that we can operate on them and still keep all the resolution of the very small differences that may exist in these binding sites in terms of affinity and specificity. And when we apply this to the mouse and the human cortex in a completely independent fashion, so 2 different data sets, 2 different models, and 2 times this TF-MINDI approach, we now see a t-SNE of all these individual sites. So each point here is not a cell, it is a binding site in an enhancer, and all the predicted binding sites are combined across all the enhancers for all the cell types of the cortex. And you can see if you do this for human and mouse, how similar the space of seclets is. So it's remarkably similar. Nevertheless, these are not in their orthologous positions. So this is what we expect, that by the differentially expressed genes which we observe between human and mouse cell types, this must underlie moving of enhancers, de novo generation, turnover of enhancers and binding sites, which is indeed the case. And I think with this, we have a good handle on the actual underlying building blocks of the enhancers in the cortex in this case. We also applied this to earlier neural development in the neural tube using organoids and a 4-week human embryo for which we have a multi-ohm atlas of the head, trained different models. So now we can compare the robustness of these models in the same genome in human for 2 different data sets. And using VISTA enhancers, which is similar to armamentarium, but with lower resolution here, with many enhancers of the neural tube, we can again assess how good these models are. These are the 2 models on the embryo and on the organoid. And again, we find that the space of all the binding sites, of all the enhancers, of all the cell types in the neural tube, derived from the organoid model and from the embryo model is highly, highly similar. And this provides confidence into this code, which allows us to build these mechanistic models further. Next, I wanted to discuss a bit about how we can use these models to assess individual variation. For that, we have built large atlases supported by aligning sciences across Parkinson's of 2 brain regions, cingulate gyrus and substantia nigra, across many donors, many human donors. Besides the multi-ohm data, we also expanded particularly the single-cell attack site to get really good models. So you see that we have many more attack cells and RNA cells to get very good crested models trained here on the reference genome and on the pool across all the donors first. Now we can use these models first. Then for all these donors, we also sequence the whole genome with Oxford Nanopore sequencing so that we have all the SNPs, indels, and structural variation, as well as phase genomes for all these donors that are matched with the multi-ohm data. And because it's ONT, we get for free also the bulk methylation patterns here so that we can link per haplotype which methylation changes occur together with SNPs, indels, or deletions. And now we can go to every variant, SNP, indel, SV, and provide the sequence of the different haplotypes to the model and assess how different the predictions are. So there's a SNP here that's predicted to increase accessibility in the alternative, where the position here is a G, in oligodendrocytes. And with this prediction, we can do this for all the 7 million SNPs and 800,000 indels across all the cell types. We can now go to the data, to the atlases per individual, and assess how good these predictions are. So one way to do this is by chromatin accessibility QTLs, where you see here an example, this one that I showed. The model predicts that the alternative G position is increasing accessibility, so we can stratify all the individuals, and you see indeed, if you are GG alternative homozygous, you have a high peak, and if you are reference A, you have a low peak. You can see some examples here, no peak in AA, high peak in GG. And interestingly, the heterozygous AG are in the middle. And so for those, we can do the second metric. All the heterozygous individuals, we can assess allelic specific signal to see if indeed the attack peak is on the allele that is predicted to be accessible and not on the other allele, which is the case here. We get for free again the MQTLs because we have methylation And so we see here that indeed this SNP, which is increased accessibility, decreases in methylation. Luckily, and what's nice to observe is that these two metrics, the QTL across individuals and the allelic imbalance within the heterozygotes, are highly concordant. So it's a very robust metric. And also are highly concordant with the methylation QTLs. Even though the methylation is on bulk, the signal here is, I would say, remarkably anti-correlated. So when you have an attack peak on a SNP, you have hypomethylation and vice versa. And then comes the result. So now we tested whether the crested model predictions based purely on the sequence, including or excluding the SNP or indel, are concordant with the actual changes we observe in these people. And this is the case. So we get, depending on the threshold, more than 90% concordance, both for allelic imbalance and for CQTL, leading to a very high confidence set of more than 20,000 variants that are enhancer variants. We can go back to the TF-MINDI map. This is all the binding sites of the cingulate cortex in human across all these millions of cells trained in the model, and then get the binding sites and project on this which binding sites are created or destroyed upon these SNPs. And as you can see, SPI1 is there for microglia, and we get some interesting things, including AP1 for the upper layer IT neurons, which change accessibility between individuals. Some of these, about 15% of these enhancer variants, we can also link to a change in gene expression, because we have RNA-seq data across all these individuals too, as is multi-ohm. We have the eQTL as well. So the chromatin accessibility QTL is correlating and concordant with the eQTL. And so this generates a nice set of enhancer variants to gene expression links, even up to 1 megabase distance, that we can now ask, can we predict this using these large models? So Informer, Borzo, Borzo Prime, Alpha Genome, we'll have a talk by Ziga as well. And we fine-tuned these type of models, the Borzo model using Scooby from the Gagneur lab, where we fine-tuned this model on the singular gyrus.
Actually, Stan,
yes?
you have 1 minute remaining.
Okay, I will finish.
Okay.
This predicts gene expression well, but some of the variants can be predicted by this model, but many cannot, as you can see here. And so this gives a good metric to improve these models. In the last 30 seconds, just a quick thing that we are also building gene regulatory network models from these large sequence to function models, so that we can actually get to the trans environment and apply to human and mouse cortex. We find similar transcription factors now that are activating these enhancers, which can be co-embedded between human and mouse in this enhancer embedding, so that we can see which ones are conserved and which not, leading to conserved and divergent gene regulatory networks, which I can discuss with you offline. I will skip the conclusions. Thank everybody. Thanks, Stan, for the neural tube human embryo data, Nelson and Trygve for the nice collaboration and hosting our student Niklas, Sarodja, and our local collaborator Pierre, and all of you again for the opportunity here. Thank you.
Great. Thank you so much for the wonderful presentation. We take a couple of questions and then we move on to the panel discussion. Doc, you have a question?
Hi, it's Doug Kim from the NIH. I wanted to go back to the M1 dataset prediction. I wondered if you could comment, are there any features of cell types that sort of jump out that the model has a hard time predicting enhancers for? What are the hard cases for the model in terms of the cell types and enhancers?
Yes, there is first of all, there's a relationship between the number of cells that are in each class that is used as a training data. So that is a trend that we observe. And actually, because there's more and more of these data sets and models, we can start having some kind of a rule of thumb that above 100 cells is certainly needed. 200 cells is probably better. If you only have 50 cells in a cluster, the pseudo bulk attack profile for training this model is too noisy and not good enough. There are also cell types that, even though they have enough cells, are not performing as well, because for each cell type separately, we can calculate precision and recall on left out data or on the armamentarium. And there I really don't have an answer for actually at the moment. So this is very interesting to me too. And this is a new challenge, I think, a new project to, after the, let's say, low-hanging fruit, finally this is working. we can now go and study where it doesn't work, where it fails, cell types that fail, or within cell types, certain enhancers which are correctly, which are validated and found to be active, which the model doesn't predict well, and go over these cases. For this, a little bit more data would be nice, but it's of course tough to get all these AAV in vivo enhancer testing done. But there's some cell types which don't have a lot of enhancers that are, on target, off target, mixed. And so that is also still a little bit limiting.
All right. Thank you.